let's continue our discussion of blood vessel anatomy by talking about veins. Veins typically outnumber arteries. You have quite a few more veins than you do arteries, and they have a larger lumen compared to arteries. At any given time, about 70% of your total blood supply in your whole body can be found in veins. Now y'all, 70%. Remember, you've got over 5 liters of blood in your body. 70% of that can be found in your veins at any given moment. Now this does include both your systemic and your pulmonary veins, okay? But this allows your veins to act as blood reservoirs. So if we need to start pumping some extra blood, we don't have to create it from scratch. That would be silly and time consuming. Instead, we can just start moving it back through your circulatory system instead of just kind of letting it sit in your veins for a little while. Veins typically have much thinner walls compared to arteries, fewer elastic fibers, and less smooth muscle compared to arteries, but we did mention that they have larger lumens than arteries. We also classify veins based on their size, just like we did arteries. So venules are your smallest veins. Your post-capillary venules specifically are going to drain the blood from the capillary beds. These are composed of basically just endothelial cells and a little bit of connective tissue. They are thin enough to allow for exchange of material through your interstitial fluids. As you get a little bit larger, larger venules, and then eventually become a legitimate vein, the 3 tunics that we have mentioned become much more distinct. Now, many of your veins also have valves. These venous valves are technically extensions of your tunica intima, and they function very similarly to the valves that we found in the heart. They are going to prevent backflow. These are especially prominent and numerous in the veins of your legs, because your legs are at the bottom. I know that sounds weird, right? Especially when you stand up. Your blood pumping, we have to overcome gravity. And bless your little veins, they don't have that thick layer of smooth muscle that we've mentioned. And so any blood that we can get up over a valve, we'll close that valve so at least the blood can't flow backwards. Now, we can compare anatomically our arteries and our veins. So we mentioned elastic and muscular arteries. Elastic arteries obviously have more elastic fibers. Muscular arteries have more muscle fibers. And then your arterioles, we said they are thinner. They do still have smooth muscles though as well, just not quite as many. And then venules, again, still very thin, even fewer smooth muscle cells compared to arterioles. The veins, we have increased smooth muscle content a little bit, but this is still not nearly as thick as in the arteries themselves, and then we can see that valve in this vein as well. Now those valves, again, we're preventing backflow, just like we did in the heart. These smooth muscles in the vein, we can contract these through sympathetic stimulation. This helps with the venous return, but again, especially in your legs, we've got to overcome gravity, y'all. So as we move up past these valves, we close the valve, and so at least the blood can't flow backwards. But that's really not going to be enough to get all of that blood back up towards your heart. So we need a little bit of help. We're going to get that help from your skeletal muscle pumps and your respiratory pumps. Your skeletal muscle pumps, basically the skeletal muscle in your lower legs that surround your deep veins. Okay, now this really only applies to the ones like deep in your legs. Every time these skeletal muscles contract, they squish, right? We're going to contract. We're going to shoot that blood up, and as it goes past this valve, we can close this valve, and now we've got more blood a little bit higher in your leg, okay? Again, this really only applies to these deeper veins. Any of your superficial veins on the outside are obviously not going to benefit from those skeletal muscle pumps. Your respiratory pump helps move blood through your thoracic and your abdominal cavities. The pressure changes that happen within these 2 cavities during ventilation helps propel the blood through the venous system as well. So between these 3, your valves, 4, the valves, the smooth muscle, the 2 pumps, we do okay with venous return. Now, varicose veins, a lot of people have heard, maybe seen, maybe even suffer from varicose veins. Varicose veins are really just dilated and bulging veins. A lot of times they're actually kind of hard to the touch as well. These tend to be the superficial veins of your lower limbs. Basically anything that decreases the rate of return of venous blood. So for example, pregnancy, standing up for long periods of time, abdominal obesity, all of that's going to cause blood to pool in the veins of your lower limbs. And because the superficial veins don't have that support of those skeletal muscle pumps, the excess blood volume stretches those veins out. It causes them to dilate, enlarge, bulge out, and then we can see them beneath your skin a little bit more visible than we normally would. High pressure in your abdominopelvic cavity during defecation or childbirth will also decrease the return of venous blood from your anal veins, which are also not well supported by the surrounding tissue. So these veins can also become weak and dilated, and that's how you get hemorrhoids, y'all. So just in case you ever wondered what a hemorrhoid was, now you know. There you go. Just in case you've never seen varicose veins, a lot of times people call these spider veins, but again, usually found on your lower legs. And then hemorrhoids can be both internal or external. You've got anal veins in both places. So now you know all about hemorrhoids, y'all. You're welcome. Okay, capillaries. These are extremely thin vessels. We're talking really just endothelial cells and a little bit of basal lamina. You can also find these little cells are called pericytes. So peri, means kind of around these cells that are around your endothelial cells. These aren't found everywhere, so you only find these in some particular capillaries. These have contractile filaments within them, and they help control the blood flow through the capillaries themselves. We said these were really thin, right? Extremely thin vessels. Like how thin, Miss Davis? Y'all, some of these, 1 to 3 endothelial cells around. Like that's it. That's it. Look, 1, 2, 3. That's the whole capillary. That's all you get. 3 cells. Okay, like that's pretty thin. Okay, got a little pericyte here. Okay, now this is a picture of a real capillary. I think this picture is really cool, y'all. Look at this. Okay, so these little darker lines. Okay, that's the capillary itself. These little red things. These are your red blood cells. These things are so skinny. 1 red blood cell at a time. These are single file because there's literally just no room for them to go side by side. A single file line, y'all. I just think this is a cool picture. Anyways, this is where capillary exchange happens. So this is thin enough for the blood or the nutrients and oxygen and stuff in the red blood cells and the blood itself to quickly diffuse into the surrounding tissue, and we can take up all the waste products back into the blood as well. So speaking of diffusion, let's remind ourselves what diffusion and osmosis are. So some of your capillaries have these really small pores. They're called fenestrations. I don't really know why the word pore wasn't good enough, but we use fenestration instead. We use these little pores, these fenestrations, to allow for diffusion and osmosis of substances. So remember, water is the diffusion of water, okay? And then diffusion, we can move small substances based on concentration gradients. And we're talking small things like monomers, monosaccharides, amino acids, things like that. So we can move these particles, these substances, through these pores in the endothelial cells. If you are a lipid soluble or a hydrophobic substance like oxygen, carbon dioxide, certain lipids themselves, we don't necessarily need a pore. We can diffuse right through the plasma membrane of these endothelial cells themselves. And then we have one more option. We could do transcytosis. So basically transcytosis is endocytosis and exocytosis. So we take in something, we endocytose it on one side of the cell, we move it to the other side of the cell, and then we do exocytosis, and it exits the cell. That's not bad, right? We take it in on one side, it exits on the other side. So that's transcytosis. So let's look at our little picture just to make sure we understand these differences. So we've got these pores, these fenestrations, okay, and substances are moving between these fenestrations. Okay. Some substances can instead move right through the plasma membrane itself. That's a different type of diffusion. And then here we have transcytosis. We're taking something in on one side and it is exiting on the other side. So we have both endo and exocytosis here. So we have different ways to do exchange between capillaries and surrounding tissues. We also have different types of capillaries. Each of these, you can tell right away, looks quite a bit different. Continuous capillaries are going to be held together with tight junctions. You're really not going to have a whole lot of things leak out of these capillaries. Fenestrated capillaries, with those fenestrations, you do have more substances that are able to move through those pores, and we're going to do more exchange that way. And then sinusoidal capillaries, it's almost like a laughable thing. This doesn't even really look like a capillary because there's these giant spaces in between these endothelial cells, and these fenestrations are even larger. So it will not shock you to learn that these are the leakiest of the capillaries. We can allow large substances to cross the capillary walls, perhaps even like an entire white blood cell or something can get out of a capillary into a tissue. Now, the blood flow that takes place at the capillary beds, we call this microcirculation, just because it's such a small setup. We have both true capillaries where things are exchanged, and then we have a small central vessel that the capillaries branch off from. You can see that we bring in oxygenated blood in our arteriole, and we're going to move it through the central vessel, and it branches off into the capillaries themselves. As we move from left to right, we are using up that oxygen, we are picking up waste products, and we are returning through that venule. We also have precapillary sphincters, both between the arteriole and the central vessel, as well as between the central vessel and the capillaries themselves. This allows us to regulate how much blood is actually flowing into the capillaries for gas exchange, or substance exchange, I should say.